Hey everyone, we hope you're having an amazing day. Today we're going to be talking about proteins. So proteins are one of 4 macromolecules, others being lipids, carbohydrates, and nucleic acids. Next we're going to talk about the composition of proteins a little bit. Proteins are made up of different amino acids put together. So therefore the amino acids are dubbed the monomeric unit. Amino acids are composed of the alpha carbon bonded to a hydrogen, a carboxyl group, a amino group, as well as the R side chain. Now, each amino acid has a unique R side chain, which gives it its identity. When 2 different amino acids come together and react, it forms a peptide bond, and this releases a water molecule. The peptide bond forms between the carboxyl group of one molecule of amino acid and the amino group of another amino acid, as you can see here. Now, once we do form a peptide bond between 2 different amino acids, we form what is called a polypeptide, or a peptide in this case. If you look over here, it has a carboxyl end as well as a amino end. This is a carboxyl end because the carboxyl group is free. It only has the bond to the carbon. It does not participate in the peptide bond. In this case, this is a amino end because the amino group does not participate in the peptide bond. The carboxyl does. Therefore, this is the amino end and this is the carboxyl end. Next, we're going to be talking about the primary structure of a protein. The primary structure of the protein is a linear structure. Now, it is basically the sequence of amino acids that make up the protein. If you Google the structure of a protein that you've learned so far, you'll likely get something that looks like this or this. The primary structure is what makes these. What this means is, if you picture this as tangled earbuds, right? And you untangle it and straighten it, you get the primary structure. It is the sequence of amino acids that make up the protein. Next, we're going to talk about the secondary structure of a protein. Now, there's 2 types of secondary structures for proteins. There's the alpha helix and then there's the beta sheets. Let's talk about the alpha helix first. As you can see here, the alpha helix, it looks like a coil, right? And it retains this shape due to hydrogen bonds between an amino acid and another amino acid 4 residues away. What this means is every 4 amino acid, there is a hydrogen bond with its 4th complementary amino acid. So as you can see here, this is the 10th amino acid. The Ahmed group has a hydrogen bond with the 6th amino acid's carbonyl group. This is a better picture to see how the hydrogen bond looks. The amide group's hydrogen forms a hydrogen bond with the carbonyl's oxygen. Another thing to note is the side chains, the R groups, are all facing out while the backbone is on the inside. And the alpha helix is always a right-handed coil, which means if you stick your right thumb up and make a fist with your hand, your fingers show the direction of the alpha helix. Now if you look here, this is a beta sheet. Beta sheet is basically when the polypeptide folds on itself. Again, this also forms hydrogen bonds, except here it's not 4 amino acids away, it's usually further. The carbonyl group of one amino acid forms a hydrogen bond with the amide of a different amino acid along the same polypeptide chain that has folded on itself. Now, it can either be parallel or anti-parallel, but the anti-parallel is more stable. Next, we're going to talk about the tertiary structure of a protein. The tertiary structure is the overall 3D form of one polypeptide chain. So if you look here, it includes the alpha helix and beta sheets. If you have a sequence of amino acid and you let it interact with itself, you get the tertiary structure. A good way to think about it is the tangled earbuds scenario. Now, in tertiary structure, it involves the interactions between different amino acids R groups, and it also takes into account hydrophilic and hydrophobic regions. Hydrophilic regions are going to want to be together, while hydrophobic regions are going to want to be together. Now, when it comes to secondary structure, as we saw, it's only interactions between the backbones, not the R groups of the amino acids. The R groups only face out, for example, in the alpha helix, while in the tertiary structure, the R groups can interact with each other. Lastly, we're going to talk about the quaternary structure of a protein. Now, a protein only has a quaternary structure if it has more than one polypeptide chain. Now, a good example of a protein that has a quaternary structure is hemoglobin. Hemoglobin has 4 different subunits, 4 different polypeptide chains that make up the overall protein. In this case, this protein has 1, 2, 3, 4, 5 different units that make up the protein. It's basically different tertiary structures interacting with each other. That makes up the quaternary structure. And note that not every protein has a quaternary structure. If they're made up of one polypeptide chain, then it only has a tertiary structure. Lastly, we're going to talk about the 3 primary functions of proteins. Proteins provide structural support. Proteins catalyze different chemical reactions, as we can see here. And proteins also allow the transport of molecules in and out of the cell. Now, coming back to this point, proteins can act as enzymes to speed up different reactions by lowering the amount of energy required to make the reaction go. So if you look here, maltose binds to the enzyme's active site. The active site is basically a pocket within the tertiary structure or quaternary structure of a protein in which reactants can bind to. Note that active sites are very unique to different reactants. It doesn't just bind to any reactant. The reactant has to have certain properties that allows it to bind into the active site. Maltose binds to the active site and is broken into 2 glucose molecules. Note that reactants are not consumed. It simply just binds and then is let go. In this case, it's broken into 2 glucose molecules. Lastly, another thing to note is the fact that enzymes does not change the energy of the overall reaction. It simply just speeds it up.